This is me Arju here and I welcome you to my channel Mr. Teeth, where your dental concept get dissected. So this is our part 2 video of the pulp, and in this video we will be discussing the structure of the pulp or the components of the pulp. So in this video we are gonna be studying the cells of the pulp. Okay, so the components means different kinds of cells are present in the pulp, and we will be discussing with each type of cells that is present in the pulp. Similarly, we will be discussing with the intercellular substances. Okay, and in the previous video of the pulp we have discussed with the histological zones of the pulp, which was one of the most important topics in that video. Okay, and it is important for your university level examination too. Okay, so most of the time they used to ask in the paper. Okay, so write the histological zones of the pulp with the proper diagram. So you should have to write the theory and you should draw the diagram properly. But in this video we will be discussing just with the components of the pulp, and today's video is properly a theory based. Okay, many diagrams are not present in this, but just small structures are there that you should remember that. Okay, so without delaying much, let's get started. So in this video we will be discussing the structure of the pulp or the component of the pulp. So in the component of the pulp we have three major components. One is cell, another is intercellular substances, and another component is a blood vessel, lymphatics, and the nerves. So in the cell section we have properly 4 types of the cells. Okay, so in those 4 types, okay, so the first type is a formative cells, and in the formative cells we have odontoblast and the fibroblast. Similarly, in the reserve cell we have undifferentiated mesenchymal cells, okay, which is a bit similar with that of the pulpal stem cells. Okay, so they are quite of same type of the cells. Okay, and another is a defense cell. So in defense cell we have neutrophil, basophil, eosinophil, lymphocyte, blood monocyte, macrophage, dendritic cells, and the plasma cells. Okay, similarly in the intercellular substances we have major 2 component, which is a fiber and the pre-collagen fiber. Okay, fiber or the pre-collagen fibers, and another component is a ground substance, and another third component is our blood vessel or the nerves or the lymphatics. Okay, so we have major 3 components of the pulp. First component is of cell, second component is of intercellular substances, and third component is of blood vessel, lymphatics, and the nerves. So in the cell we have properly 4 different types of the cell. One is formative cell, another is reserve cell, another is defense cell, and the pulpal stem cell. And in the intercellular substances we have major 2 components. One is fiber or the pre-collagen fiber, and another is a ground substance. Similarly, our major third component of this pulp is blood vessel, nerves, and the lymphatic. So first we will discuss with the formative cells. First we will discuss with the intercellular substances. Okay, so in the intercellular substances, as I have told, it have major 2 component. One is fiber or the pre-collagen fiber, and another is a ground substance. Okay, so now intercellular substances is a dense and the gel like structure. Okay, like in nature, and finally granular to the fibrillar. Okay, so it is easy to understand. So the pulp have intercellular substances. Okay, so intercellular substance means the substances that is present between the 2 cells. Okay, so if this is one cell and if this is another cell, okay, so the space that is between those 2 cells is the intercellular space, and in those spaces there is presence of the substances, and that substances are called intercellular substances. So intercellular substances, it is quite of dense and the jelly like, okay, which is granular to fibrillar in structure. Okay, which is finally granular to the fibrillar in structure. And in the fiber section, okay, so this fiber is one of the most important. So they can ask you in a fiber what sort of fiber is present in the intercellular substances of the pulp, then at that time you should tell in fiber section. Okay, so we have particularly collagen fiber. Okay, so in collagen fiber we have a type 1 and type 3 type of collagen fiber present here. Similarly, in the cross striations are also present in those collagen fiber, and it is present at the distance of 64 nanometer. Okay, similarly the length of the collagen fiber ranges from 10 to 100 nanometer. Okay, so this is one of the range, it is a average. Okay, so it may be smaller than 10 or maybe greater than the 100. So it is just a range of the length. Okay, so you can remember it. So cross striations present at the 64 nanometer, and the range of the length of this fiber is 10 to 100 nanometer. And the fine fibers, okay, so the fine fibers are called fibrillin, which is one of the important terminology of this fiber. Okay, so you should remember what is fibrillin. Okay, so they can ask you in a Viva. So the fibrillins are the fine fibers that are present in the intercellular substances of the pulp. And the major another thing that is present here is this. Okay, so the degradation and the down regulation of this fibrillin is helping in the release of the TGF beta. Okay, so it releases this TGF beta, and this TGF beta helps to form the mineralized tissue barrier. Okay, so it forms the mineralized tissue barrier, which is important to remember here. Okay, and after that, in the amorphous ground substances, okay, so with this fiber, okay, so with this fiber also we have ground substances, and this in amorphous ground substances we have different kind of biochemical substances, like we have glycosamine glycans. Okay, so gags, so they are hydrophilic and they form the gel-like structure. So this gag is also present in our development of the tooth. Okay, so there I have discussed with this glycosamine glycan, and this glycosamine glycan helps to form the stellate reticulum. Okay, star like reticulum there. Okay, so I think you have remembered that, and that was important there. So similar here, the glycosamine glycans is present here. Similarly, this chondroitin sulfate is present here, hyaluronic acid is present here, glycoproteins, water, and different kind of salts are also present there. Okay, small amount of the minerals. Okay, salts, minerals. Okay, so minerals are particularly present there. Okay, so the main thing that you should take from these intercellular substances is that, okay, so you just remember there are two major components of these intercellular substances. One is a fiber or the collagen fiber, and another is a ground substance. So in the fiber or the pre-collagen fiber, so they are made up of the collagen, and we have particularly two type of collagen fiber, type 1 and type 3. Okay, and cross striations are present at the 64 nanometer distance, and the length of the fiber is 10 to 100 nanometer. Similarly, those fine fibers, okay, so the fine fibers of this pulp of these intercellular substances are called fibrillin, and with the degradation and the down regulation of the fibrillin, they release the TGF beta, and those TGF beta are responsible for the formation of the mineralized tissue barrier. Okay, and after that, in the ground section we have different kinds of biochemical substances like glycosamine glycans, chondroitin sulfate, hyaluronic acid, glycoprotein, and the water. Okay, so you should remember all those things from these intercellular substances. Now we will move to the major topic, that is of cell. Okay, so we will start with the formative cell. So in the formative cell, as you can see here, our first cell is the odontoblast. Okay, and I think you can teach me about this odontoblast. Okay, so as I have discussed in the dentin properly about this odontoblast. Okay, so some important more things, okay, are present here, so you can relate all those points from both the chapters. Okay, so our first cell is odontoblast, and in first point, okay, as I have written here, it is derived from the peripheral cells of the dental papillae. Okay, which is very easy to remember, as you have, if you have watched the video of development of the teeth, there I have properly discussed. Okay, so this dental papillae give the two structure, one is pulp and another is a dentine. Okay, and the dental sac or the dental follicle gives other structure like cementum, okay, periodontal ligament, alveolar bone. Okay, our second point is that it is a second most prominent cells of the pulp. Okay, and after that, the part of the odontoblast, we have cell body, neck, and odontoblastic process. Okay, so I will draw a schematic diagram here. So just think this is a odontoblastic cell. Okay, and this is constricted at the neck portion, and this is a process of the odontoblast. Okay, and here is the nucleus. Okay, so this is a proper odontoblastic cell. Okay, so it is the parts of the odontoblast. So just see, this is a body, okay, and this is a neck, okay, which is constricted, and this is the odontoblastic process. Okay, so this is a body, this is a neck, and this is odontoblastic processes. So B and O you can remember by this way, or it is easy to remember this picture itself. Okay, so the cell body, it resides in the pulp adjacent to the predentine. Okay, so as I have discussed in dentine chapter, so the adjacent to the predentine there is a layer of the odontoblast. Okay, so in the pulp we have odontoblastic zone. So in that odontoblastic zone we have this odontoblast cell body present there, and after this cell body we have neck. Okay, so the neck is a structure that is constricted. Okay, and it is a constricted portion of the cell body towards the dentinal side. So that neck is constricted to the diameter of 3 to 4 nanometer. Okay, you can remember this. And after that, in the upper neck, so there is odontoblastic processes. Okay, and those odontoblastic processes are this, and they run in the dentinal tubules. Okay, so we have particularly three major parts of the odontoblast. One is a cell body, another is a neck, and then is a odontoblastic process. So the cell body resides in the pulp, that is just adjacent to the predentin, and that cell body just get constricted towards the dentinal side, and that is called the neck, and it is constricted to the diameter of 3 to 4 nanometer micrometer. After that, it just move in the dentinal tubules. Okay, and that is the dentinal that is the odontoblastic process. Okay, and I have discussed properly about this odontoblastic process, dentinal tubules, in the dentin chapter. Okay, so you can go through that. After that, in the shape section. Okay, so in the shape, okay, so this is also very important for the Viva, especially Viva examinations. So they can ask, okay, so what are the different shapes of the odontoblast at different areas of the pulp? So at that time you should tell that at the coronal area it is a quite of columnar type. Similarly, at the cervical area it is a cubital, and at the root area it is flattened and ovoid. Okay, so coronal means crown portion. Okay, so in the crown portion of the pulp it is quite of columnar, in the root portion it is quite of flattened ovoid, and at the cervical area it is quite of cuboidal. And after that, in the next point, 5 to 7 micrometer in diameter and 25 to 40 micrometer in the length. Okay, so this is a dimension you can remember. So 5 to 7 micrometer in the diameter and 40 micrometer in the length, 25 to 40 micrometer in the length. After that, shape of the odontoblast reflects the functional activity of the cell. Okay, so this is also one of the most important thing to remember from this odontoblast. Okay, and I think I haven't told this before. Okay, so this shape of the odontoblast reflects the functional activity of the cell. So what is the shape of the odontoblast? It signifies its functionality. Just like there are particularly two types of the odontoblast based on the function. One is active cell and another is a non-active odontoblast, non-active phase. Okay, so in the active or the secretory phase, there is an increase in number of endoplasmic reticulum, Golgi apparatus, and the secretory vesicles. Similarly, the resting or the non-active phase means cells are flattened like cytoplasm, condensed chromatin, and the decreased number of the endoplasmic reticulum. Okay, so just you should remember here the thing is the main thing that you should remember from this. Okay, is that there are particularly two forms of odontoblast. One is active and another is a resting or the non-active. So in the active form we have a so large number of cell organelles present there. Okay, so we can see different numbers of the endoplasmic reticulum, Golgi apparatus, secretory vesicles present in the active cells of the odontoblast. Similarly, in the non-active phase of the odontoblast we can see the flattened little cytoplasm. Okay, so little condensed cytoplasm is present in that non-active phase with the condensed, so little cytoplasm with the condensed chromatin and a decreased number of ER. Okay, so there is decrease in the organelle number, and also there is a little cytoplasm in comparison to that of the active odontoblast, and we have condensed chromatin. Okay, so we have condensed chromatin, and there is a non-functioning of the nucleus. Okay, so there is not formation of any kind of protein. Okay, any kind of secretory vesicle is not forming in the odontoblastic in this phase of the odontoblast. Okay, after that, the functions of the odontoblast I have mentioned here. So it helps in the formation of the dentin, which is of sure. After that, it helps in the maintenance and the repair of the dentin. It is also important. Supply of the nutrition to the dentin. Okay, so the every nutrition it comes from the pulp. Okay, so the odontoblast and odontoblastic processes, okay, it helps in supplying the nutrition to the dentin. Okay, and it helps the sensation of the dentin too. Okay, so the dentinal fluid, okay, dentinal lymph, they are also formed in the dentin by the help of the odontoblastic processes. Okay, so they are the plasma infiltrate. Okay, as I have told in my dentin video, so they are the plasma infiltrate, dentinal fluid. Okay, so you should remember all those points. And after the odontoblast, okay, so we will discuss with this fibroblast. Okay, so this fibroblast, and just remember all those important points. Okay, so I have mentioned everything here, and I guarantee you after knowing all those points you will be properly prepared in this chapter. So just follow my instruction, follow those notes and study well. Okay, after that, in fibroblast, it is also the most prominent cells of the pulp, and second most prominent cell of the pulp is the odontoblast, as I have mentioned here. After that, the content of the fibroblast We have abundant rough endoplasmic reticulum here, mitochondria and other organelles that indicates the cells are active. Okay, so see, it is similar with that of the active phase of the odontoblast. Okay, so there is lot more presence of the rough endoplasmic reticulum, mitochondria, organelle, okay, that indicates the cells are active. After that we have in young pulp and in the older pulp. Okay, so there is a different in the structure, there is difference in the structure of the fibroblast in young pulp and in older pulp. So in the young pulp, okay, we have typically stellate shaped fibroblast, and in the older pulp we have rounded or the spindle shape. Okay, so stellate means, as we have discussed in the, okay, so it is quite of star like. Okay, so this is again connected to the another, so it is quite of star like. Okay, so this is stellate type of shape, okay, that is present in the young pulp. And the rounded or the spindle shape means, okay, so this spindle shape means just like this. Okay, and there is nucleus. Okay, so it is in spindle. Okay, so cell divide and are active in the protein synthesis, and short process fewer organelles termed fibrocytes. Okay, so remember here, so we have different two types of fibroblast. Sorry, we don't have two types of fibroblast, but we have difference in the shape of the fibroblast and their function with the age of the pulp. So in the young pulp we have typically stellate shape, and in the older pulp we have rounded or the spindle shape. And the young pulp we as they are more functioning. Okay, so the cells divide. Okay, so the fibroblast cell, it divides in high speed. Similarly, they are active in the protein synthesis. Okay, so they are active in the protein synthesis. After that, in the older pulp, they are rounded or the spindle shape, and the short processes or the fewer organelles, and they are termed fibrocyte. Okay, which is important to remember from this. Okay, so the short processes, fewer organelles termed fibrocyte here. After this, in the function section, okay, so in the functions, as I have mentioned here, so the fibroblast, okay, they forms and maintain the pulp matrix. Okay, so the particularly simply just okay, if you don't remember all those functions, just remember that fibroblast are the thing that secretes the pulp matrix, different kinds of fiber. Okay, as the fibers are present in the pulp matrix, so they particularly form the fibroblast, means fiber forming cells. Okay, so they form the fiber, as they form the fiber, they form the matrix. Okay, so remember like that. Okay, so it forms and they maintain the pulp matrix. Similarly, same thing, matrix synthesis, collagen fiber synthesis, same thing. After the degradation is the of the matrix, so this is one of the important point here. Okay, so the degradation of the pulp, okay, which is one of the most important thing in this fibroblast. Okay, so the fibroblast have dual function, it just can secrete the pulp matrix and also can degrade the matrix. Okay, so it have dual function, one is formation also and another is degradation also. So this is one of the important thing of this fibroblast, you should remember. After that, role in the inflammation. Okay, so the role in inflammation and also in the healing. Okay, so it secretes FGF2 and VEGF. Okay, so those type of okay, chemokines. Okay, and the chemical mediators are secreted by this fibroblast during the inflammation and the healing. After this fibroblast, we have another type of the cell, and that are reserve cells. Okay, so in the reserve cells are undifferentiated mesenchymal cells. Okay, so reserve cells constitute those undifferentiated mesenchymal type of the cells, and these are the primary cells of the young pulp, but few are seen in the pulp after the root completion too. Okay, so they are the particularly cells of the young pulp. Okay, they are not present in the older pulp, as we can see in the previous paper in fibroblast. Okay, so there was a fibroblast present in the young pulp too and in the older pulp too, but these reserve cells are particularly present in the young pulp only. After that, it is larger than fibroblast, which you should remember, it is larger than the fibroblast, and it is polyhedral in shape with the peripheral processes, large oval staining nucleus. Okay, so polyhedral means, okay, so this is a polyhedral. Okay, so this is a polyhedral. Okay, and with the processes. Okay, so there is running of the processes from the edges. Okay, so the peripheral processes are also present there, and the long oval staining nucleus. Okay, so there is long oval staining nucleus is also present here in reserve cell. After that, this reserve cell, this undifferentiated mesenchymal cells are also totipotent type of cells, and totipotent means it becomes the odontoblast or the fibroblast with the need. Okay, I think you know this term, this totipotent. Okay, So totipotent cell means it can differentiate into any other type of the cell. Okay, as I have told previously, okay, we have pulpal stem cell. Okay, so stem cell and this undifferentiated mesenchymal cells quite have similar functions. Okay, so I have not written about the pulpal stem cells here, so you can just relate all those points with that cell too. Okay, so they are just totipotent cell, which is the important function of this undifferentiated mesenchymal cells. So with the need of the cell, they can just differentiate into the odontoblast or the fibroblast. Similarly, due to decrease in the number and activity in the older age. Okay, so in the older age there is decrease in the number and the activity. Okay, number and activity is decreased with the age. So in this undifferentiated mesenchymal cell, the point you should remember is that undifferentiated mesenchymal cells are those cells that are larger than the fibroblast. Okay, and in the second point you should remember that they are polyhedral in shape with different kind of processes, peripheral processes present, and the large oval staining nucleus is present in the cell. And the third point is the totipotent nature. Okay, so the totipotent nature of this mesenchymal cell results in the formation of the odontoblast or the fibroblast with the need, and in the older age it is not present, but in the young pulp it is mostly present. Okay, so these 4 points you should remember from this undifferentiated mesenchymal cell. After that, in the defense cell, so in defense cell, okay, so we have different kind of histiocyte, macrophages, okay, lymphocyte, plasma cell, dendritic cell. Okay, so the histiocyte are the macrophages. Okay, so they are the irregularly shaped cells with the short blunt processes. Okay, so they are irregularly shaped, so they can be like this. Okay, so they can be like this. Okay, so any shape, they can be of any shape. They are irregular in shape with the blunt processes. So this is blunt. Okay, so this is not pointed, no, so this is blunt. Okay, so they are in the blunt processes is present there. Short blunt processes are present there in histiocyte or the macrophages, and it exhibits the granules and the vacuoles in their cytoplasm. Nucleus size also increases, prominent nucleolus during the pulpal inflammation. So in the histiocyte or in the macrophages, okay, if you will not study this, okay, and just you will remember, just simply you remember what is macrophage. So just you can see the function there. Okay, so the macrophage is just a cell that just engulf the foreign particles into itself, okay, and just degrade those substances. Okay, so that is a proper function of the macrophage. So similar thing is here. Okay, so the macrophage, it just engulfs the foreign particle into it and degrade those foreign particle by the granules and the vacuoles that are present in the cytoplasm of the macrophage or the histiocyte. Similarly, we have larger nuclear size here, prominent nucleolus here, and during the pulpal inflammation. Okay, so the same thing is written here. And another one important thing is associated with the small blood vessel and the T lymphocyte. Okay, so which is easy to understand here and should remember this. Okay, so the thing that I have told in this histiocyte and macrophage, I will recite again. So the histiocyte or in the macrophages, just to remember, the main function of the macrophage is to engulf the foreign particle, okay, that is present in the body and just degrade those foreign particle. And similar thing is here. Okay, so they contain the granules and the vacuoles in the cytoplasm, okay, that degrades that substances after ingesting the substances, foreign particle, and after degradation, okay, so they just degrade those substances, and they are of the irregular shape with the blunt processes, and they are associated with the small blood vessel and the T lymphocytes. Okay, so this is important. So they are associated with the small blood vessel and the T lymphocyte. Okay, mostly they are seen near those blood vessel and the T lymphocyte. Okay, and at those region, okay, so the immunity is required, so they are mostly present on those small blood vessels. Okay, after that, lymphocyte. Okay, so lymphocyte, they are extravascularly present. Okay, so they are not present inside the blood vessel. Okay, as they are associated with the small blood vessel, they are not quite closely attached to the blood vessel. Okay, so they are not present inside the blood vessel, they are extravascularly present. Okay, mostly T lymphocytes are present. Okay, and round nucleus and contain many dark staining granules. Okay, so I think you have read this about lymphocyte, plasma cell, dendritic cell in your pathology. Okay, just I have written a small point. Okay, and it is not quite of important here. Okay, so I am just telling a small, so I am just reciting all those points. Okay, so the lymphocyte extravascularly present, mostly T lymphocytes are present here, and round nucleus and contain many dark staining the granules. After that, in a plasma cell, okay, so the main function of the plasma cell is to okay, secrete the antibodies. Okay, so plasma cell, it produce the antibodies. Okay, and it appears during the inflammation of the pulp, and the nucleus is small and the concentric, and it is cartwheel appearance. Okay, so I think you have I think you know this cartwheel appearance. Okay, so the plasma cell is that cell that secretes the antibody, and the nucleus of those plasma cell is of cartwheel appearance. Okay, so cartwheel means like this. Okay, so cartwheel appearance. Okay, so they appear like this. Okay, so the nucleus appears like this cartwheel structure. After that, our another cell is the dendritic cell, and it is closely related, okay, to the endothelial cell. Okay, and after that it expresses the macrophage related antigen CD14, CD68, and beneath the odontoblastic layer, okay, it is quietly present beneath the odontoblastic layer in the pulp, and it capture and present the foreign antigen to the T cell. Okay, so they are antigen presenting cell properly. Dendritic cell are the antigen presenting cell, they are beneath the odontoblast. Okay, and additionally we have polymorphonuclear cell, eosinophil, basophils, mast cell, monocytes. Okay, so they are the different kind of the defense cell. Okay, so at last I will just tell in short about this defense cell, so just you listen to me. Okay, so defense cell are those cells that are having the immunity function. So we have major histiocyte and the macrophages. So the main function you should remember about this is they just engulf the foreign particle and degrade those foreign particle inside their cytoplasm, inside the cell. Okay, by different kinds of the granules and the vacuole secretory vesicles that are present inside it, and it is of a irregular shaped cell and with a blunt processes. Okay, you should remember those 2 points, and it is quietly associated with the blood vessel and the T lymphocyte. Okay, after that in the lymphocyte, so the lymphocyte we have B lymphocyte and the T lymphocyte, and you have properly studied about this lymphocyte in your pathology, and for now just you remember it, they are extravascularly present and they contain the round nucleus with the dark staining granules inside it. Okay, so those granules contains the substances that just degrade the foreign particles. After that in the plasma cell we have cartwheel appearance. Okay, so cartwheel type of nucleus present in the plasma cell, whose main function is to secrete the antibodies. Okay, so the main function of the plasma cell is to produce the antibodies. So the lymphocyte, B lymphocyte just change into the plasma cell, particularly B lymphocyte. Okay, so they just change the plasma cell and they secrete the antibodies. After that in the dendritic cell, so we have close relation to the endothelial cell. Okay, so endothelial cells are the cells of the blood vessels. Okay, so the lining of the blood vessels are called properly endothelial cell. So they are closely related with those endothelial cell, and they are just present beneath the odontoblastic layer in the pulp, and their expression, okay, so they are particularly the antigen presenting cell, so they express the macrophage related antigen CD14 and the CD68 are present there, and it capture and present the foreign antigen to the T cell. Okay, capture and present the foreign antigen to the T cell. Okay, so those are in this dendritic cell. And in the last point, as I have mentioned, with those macrophages, histiocyte, lymphocyte, plasma cell, dendritic cell, we also have polymorphonuclear cells, eosinophil, basophil, mast cell, and the monocytes. Okay, so these are also the cells that are present in this defense cell. Okay, and I think you have studied about those eosinophil, basophils. Okay, and neutrophils. Polymorphonuclear cells means neutrophils. Okay, that have multi-lobed nucleus present in it. Okay, so I think you know about this. So with this we have completed today's video, and in this video I have discussed about the components of the pulp. Okay, so we have discussed with the different kinds of the cells and the intercellular substances, okay, and the blood vessels and the nerves. Okay, so there is a I think about the blood vessel I will be discussing in the next video. Okay, but for today we have only this much. Okay, and in the next video we will be discussing further about all those topics that are remaining here in this chapter. So after telling this much, okay, and I always tell you, okay, just go through all those notes and the note that I am providing you. Okay, so you can go through all those notes and just watch this video and read all those notes. Okay, so that will be more than enough in this chapter. Okay, for your university level examination you shouldn't have to be worried about anything. Okay, as I am providing you the proper content that is present in our bands, okay, and or any other books. Okay, so you shouldn't have to be worried in any of the topics that is present in this chapter. So go through the notes properly, okay, read all the points, and I am trying to discuss all the point in the video, okay. So once watch the video, read the notes, and be properly clearing all your concept in that chapter. So after saying this much, I will be meeting you in the next video, okay. I will catch you in the next video. Till then, stay happy, stay healthy, and tata, bye bye, thank you.